Number 150, glomerular diseases, mechanisms and classification. Now this is a relatively bigger question because there are a lot of pathophysiological mechanisms we need to discuss in this question, but should you be able to understand what's going on in this question, you'll be able to better understand what's happening in the glomerular diseases that are associated with nephrotic and nephritic syndromes, which we will be discussing in the videos to follow, for the questions to follow. But before you get into those questions specifically, please make sure that you understand the difference between nephrotic and nephritic syndrome, as that too will further aid in your understanding of the diseases that are to be discussed in this chapter. So what we're going to do is we're going to divide glomerular diseases into glomerular diseases that are occurring due to a primary mechanism, which is normally in association with an immune mediated injury, and a secondary mechanism, which we'll get to in just a bit. So stick with me, hopefully this makes sense. So the first thing is I want you to familiarize yourself with this diagram here, and so I will label this for you really quickly. Up here we've got the mesangial cells, or the mesangium. Then we've got the endothelium, following this we've got the basement membrane, and then below that we have the Bowman's capsule. But what we're specifically interested in is the visceral epithelium of the Bowman's capsule. And so the first type of immune mediated attack we are going to be discussing is a type 2 hypersensitivity reaction. So a type 2 hypersensitivity reaction. Now you'll remember from immunology, but if you don't, this basically occurs when you have a intrinsic antigen that is always found in a fixed tissue attacked by antibodies that are formed against it. So what we need to remember is that the antigen in question is within the kidneys, and there is an antibody circulating in the blood that will end up in situ forming an antibody antigen complex, and then you have immune mediated damage. In type 2 hypersensitivity, it's important to note that there are no pre-existing antibody and antigen complexes circulating in the blood. The formation of these antibody and antigens complexes are formed in situ, and the antigen is always found within the kidneys. Right, so this is the first way that you can have damage to the glomerular surfaces and these glomerular apparatus. And so to give you an example of two diseases in which this sort of mechanism occurs, is that we have the basement membrane here, and we have got type 4 collagen that is found within the basement membrane. And what happens is, so I'll just label this as type 4 collagen that is found within the basement membrane. And if you look at the structure of these collagen fibers, these type 4 collagens, we'll see that we have a globular component, and we've got the tails that make up the collagen fibers. Anyways, the important point here is that these globular components can become antigenic. So these become antigenic, meaning that antibodies that are found in the blood, circulating in the blood, will now end up binding to these non-collagenous globular components of these type 4 collagen fibers, and you will have antibody deposition taking place, which will then mediate your immune response, immune mediated damage. What's important in this case is that the antibody deposition is linear. So when you look under the immunofluorescence, you will see that these antibodies are deposited in a linear pattern against antigens that are found in the glomerular basement membrane to be precise. The second type of disease where this could occur, this sort of type 2 hypersensitivity reaction could occur, is with respect to Magellan, and I will explain what that is. So we look at the visceral membrane of the Bowman's capsule, and what we see is that the foot processes of these podocytes have a protein called Magellan, and so these two can become antigenic, and you can have antibodies deposit and form an antibody antigen complex with these Magellan proteins, and then this will initiate an immune mediated damage response. In this case, the deposition of antibodies is in a granular pattern as compared to the linear pattern we spoke about with respect to type 4 collagen antibody attacks. And so these are just two examples with respect to type 2 hypersensitivity reactions that can mediate damage to the glomerular apparatus. So now coming to another type, what can happen is, and this too, the one we're going to discuss right now too, is a type 2 hypersensitivity reaction. So once again, the second type is also a type 2 hypersensitivity reaction. But what makes this different from what we just discussed is that the antigens that are found, pay attention to the antigens, these antigens are extrinsic to the kidney. So these are not proteins or not any structures found within the kidney originally that can result in damage to the, rather initiate this type 2 hypersensitivity reaction. And then the part that is similar though is that you still have antibodies that are circulating in the blood, and so when these antigens that are extrinsic to the kidney end up depositing within the glomerular apparatus, depositing in the glomerular apparatus, what can happen is that you can have once again an in situ formation, of antibody antigen complexes. Please note once again that these antibody antigen complexes are not preformed, and they are formed at the level of the kidney right then and there. Hope that makes sense. Now what we need to discuss, another thing, obviously if we're talking about these extrinsic antigens, we need to know what these extrinsic antigens are. And so these can be endogenous antigens, and this is normally DNA fragments. These are normally DNA fragments, or they can be exogenous deposition into the kidney, meaning these are antigens that come from the outside. These could be toxins, these could be drugs, they could be microbial in nature as well. Anyway, the point being that these antigens that are extrinsic to the kidney are deposited within the glomerular apparatus. The next thing you need to understand is the locations where these depositions can take place, and according to that the nomenclature and the terminology stands. So if you have deposition within occurring within the mesangial cells, so somewhere here, this is an intra mesangial deposition. If they deposit within the subendothelial layer, so below the endothelium, these are subendothelial deposits. If the antigens are to deposit within the basement membrane, these are intramembranous. So intramembranous deposits, and if they deposit below the basement membrane, these are subepithelial deposits. Please remember the difference between subendothelial and subepithelial deposits. And that's pretty much it. And as it's quite normal to understand, quite easy to understand, is that the larger the antigen is, the more likely it is to deposit in the intramesangial and the subendothelial layers, whereas the smaller it is, the more easy it is for it to pass through the layers of the glomerular apparatus and end up somewhere in the subepithelial layer. And so the smaller it is, the more deeper down it can go, and the larger it is, the more likely it is to just be stuck within the superficial layers of the glomerular apparatus, right? And as a whole, depending on where these antigens are located, or where they're deposited rather, location of deposition normally drives the impact and the progression of the disease. And so that's that. Now then, coming to type 3 hypersensitivity reactions. So type 3 hypersensitivity reactions, this is where you have circulating, so these are preformed now, circulating preformed immune complexes that deposit into the kidney and then mediate damage. And since these complexes as a whole are relatively large, the most common area of deposition is usually subendothelial. And this is normally seen, type 3 hypersensitivity reaction normally seen in, for example, post streptococcal glomerulonephritis. More on that in the questions to follow, but that is a basic overview of some of the important hypersensitivity reactions that are associated with causing glomerular damage. Then, what are some of the other causes that can cause glomerular damage and are still classified as primary? You can have direct cytotoxicity by antibodies. So these are your mesangial cells. And what you can have is that you can have antibodies that are formed against these mesangial cells specifically, but they don't bind onto and attack the mesangial cells. Like normally when you have primary mechanism, you can have, when you have an antibody that is attached to a antigen, it initiates the complement mediated destruction. In this case, these antibodies directly induce destruction of the local tissue at hand, or the local tissue cells in question, and in this case this is found in the kidney. And so these antibodies directly damage resident cells of the glomeruli, whether that's the endothelial cells, the mesangial cells, the epithelial cells, etc. Then we move on to cell mediated injury. And so once again I will draw out the basement membrane here with podocytes and their foot processes. And so pay attention here to what happens after you have a cell mediated injury. Firstly, what the cell mediated injury is that you will have a T cell, an immune T cell, a T cell that gets activated, and this reacts with basement membrane and it ends up neutralizing the charges that are found on the foot processes of podocytes. And essentially what ends up happening is that you have a flattening of these foot processes, and this as a result will now decrease the ability of the glomerular apparatus to form that selective barrier that we say is present in the kidneys and filtering out all the wastes and all the things that are found within the blood. So it decreases ability to form our filtration barrier. So that is how cell-mediated injury takes place, and one thing that can benefit you from remembering is that this activation of T cells is normally precipitated by a infection, and this normally may be an upper respiratory tract infection or also can be a UTI as well. So activation of T cells is initiated in response to an infection. And coming on to the last thing, while the complement system or the attack that is associated with the complement system rather is classified in secondary mechanisms of injury to the glomerulus, an inappropriate activation of the complement system is normally classified under a primary mechanism of damage. And so what really happens is, I'm not really going to mention all the proteins and everything in too much detail, but what you have is, you look at the complement system, you've got the C3b protein, BB protein, and the C3b protein again, that forms your C3 convertase enzyme, which initiates the further activation of a handful of other complement proteins, which in turn forms the membrane attack complex. Just remember the formation of the membrane attack complex. Now this normally is associated when you have antibodies that are targeted to specific antigens, but if you have an inappropriate activation of these enzymes, specifically C3 convertase and C5 convertase, so this bit makes up the C3 convertase and this whole molecule makes up the C5 convertase. If you have an inappropriate activation of these proteins, you can result in the formation of membrane attack complexes. And so it's normally been seen, and the reason this is still classified as a primary mechanism of injury is because you can have antibodies that are produced against C3 convertase, and these are known as properdin factors. So these properdin factors, what they do, these antibodies, is that they stabilize a bit of a mix-up. Let me correct myself really quickly. These antibodies are what end up stabilizing a factor that we call properdin factor, and it is in fact properdin factor that ends up activating the C3 convertase. So just to clarify one more time, you have something called properdin factor which ends up activating the C3 convertase enzyme, and if you have an overactivation of these C3 convertase enzymes, which downstream leads to the activation of the C5 convertase enzyme and the formation of a membrane attack complex, you can have immune mediated damage. Because this comes about by the formation of antibodies, it is still classified as a primary mechanism of injury. And these autoantibodies are in fact what stabilize the properdent factor. Hope that is clear. Now we move on to the secondary mechanism. The secondary mechanism of injury to the glomerular structures, and this is normally brought about by inflammatory cells. So this includes your neutrophils, macrophages, and lymphocytes. Or it's also brought about by other soluble factors such as reactive oxygen species, proteolytic enzymes, cytokines, and the complement system itself lastly. And so just a brief discussion about each of these. The first one being the complement system. And so we discussed, let's just go back to our basement membrane from earlier on. And we've said that we've had deposition of antibodies. Having deposited antibodies, one of the way that this reaction will further progress is the activation of the complement system. So again, I'm not really going to go too deep into the pathways of the complement system, but just really quickly, we know that the Fc region of this activated antibody will result in these proteins, the complement proteins, ending up binding onto the Fc region. We've got the C1 protein followed by the 4B, if I remember correctly, then the 2B, 3B, 5B, and then you've got 6, 7, 8, 9, and these proteins then will end up forming what we call our membrane attack complex. And this membrane attack complex will then result in damage to the endothelial cells, to the glomerular basement membrane, and to the mesangium. Once again, this is classified as a secondary mechanism because the primary mechanism of damage in this case was when you had your antibodies depositing against structures that are found in the intrinsically in the kidney in this case. Why the complement system pathway that we discussed in previously was classified also as a primary was because you had antibodies that were stabilizing the formation of this membrane attack complex, and so just keep that in mind. Anyway, that's one way this damage occurs. Secondly, what happens is, you'll remember that factor 3A, not factor but like the complement proteins 3A and 5A are chemotactic as well, and they will activate mast cells. These mast cells downstream can release histamines, they can release proteases, they can release prostaglandins, and what this ends up doing is this will further lead to hemodynamic changes, which results in an increased infiltration of inflammatory cells, specifically your neutrophils, which are chemotactic to some of these complement proteins. And so as a result, you have an elevated immune response taking place that is mediated by the complement system activation once they have recognized and bound themselves to the Fc regions of the antibodies we discussed from the previous primary mechanisms. So that's one way damage occurs. Second way is directly like some of the cells we discussed, the neutrophils, and the macrophages. We know very well that these are capable of secreting cytokines and reactive oxygen species that can mediate any form of damage. And so with respect to neutrophils, these are activated by complement protein 5A, and we know that neutrophils have the ability to release enzymes and reactive oxygen species, as do macrophages. So reactive oxygen species and cytokines, and then these two processes together end up driving endothelial and tissue damage. Now, when you have endothelial damage, we'll remember from physiology, this is our endothelial lining. We've got damage, let's say, of the endothelium here. This means we increase the likelihood of forming a thrombus in this area, and so you can end up forming a thrombus within the endothelium, which can result in further damage, and it can also occlude blood supply to the to distal to this thrombus block. And so the endothelium damage will result in makes the entire endothelial lining more thrombogenic, can further mediate damage as well. And other causes of damage that are classified under the secondary mechanism is through growth factors. And how this works is that you can have the underlying pathophysiology behind this is that growth factors that are released by mesangial cells, or the macrophages we just discussed earlier, can end up causing the proliferation and growth of these mesangial cells. And once this happens, this leads to a compression and damage to the glomerular architecture. And yeah, so coming on to the last form of damage, which is most sometimes idiopathic and not exactly too well understood, is that you can have changes that occur within the foot processes of the podocytes. We discussed previously that this can be due to T-cell mediated immune response, but in this case the underlying cause is not entirely known too well. But what we do know is that these podocyte changes results in effacement of the foot processes, and this can then in turn damage your glomerular architecture, and it decreases the likelihood of the filtration barrier working how it should be physiologically. And so the last thing we need to discuss, thankfully, is just the morphological changes that are seen in injured glomeruli. And these morphological changes aren't very macroscopic. These are normally seen under a microscope, and what we really need to discuss here are the terminologies that are used to describe these changes in the glomerulus. So if the processes we've just discussed previously end up damaging all of the glomeruli, so all glomeruli are showing pathological changes, so all glomeruli showing pathological changes. Term that we use to describe this sort of damage is diffuse. If, however, you only have some glomeruli that are involved, so some glomeruli involved, terminology we use to describe it is focal. If the entire glomerulus is damaged. Term that is used to describe it is global. And if only part of the glomerulus is damaged, the term that is used to describe it is segmental. And this is important, as you will see in some of the diseases. The naming, the nomenclature itself can give away what the disease is and what damages have taken place. So for example, we have a glomerulonephritis that is known as focal segmental glomerulonephritis. This basically means that you have only some of the glomeruli damaged and only part of those glomeruli are damaged. And so this nomenclature can help you in understanding how diffusely or how extensively the glomeruli are damaged and the underlying, and it can give you a hint as well to the underlying processes occurring as well, which you will see in future questions. And that is it for this question.